Hey everybody, I see that we're up to 57 participants on the Zoom. We're just going to give it another minute or so as people are still dialing in here. Hey everybody, we're just giving it 2 more minutes here. We're expecting a few more to log in.
Hi, everyone. We're going to get the program started today. We are so excited to welcome everyone to our first annual CTNNB1 Syndrome Research Conference. We are fortunate enough today to have in attendance families from around the world, hailing from 6 different countries. Additionally, we have doctors and researchers in attendance from 3 different countries and 4 different states here in the US. Representing such esteemed institutions as University of Washington, University of Hong Kong, Adelaide Medical School in Australia, Mount Sinai in New York, CHOP in Philadelphia, Johns Hopkins, and Children's Hospital of New York Presbyterian, Columbia University. I wanted to first thank Dr. Chung and Dr. Jacob for giving their time to speak to us today, and also to thank Seesaw for their collaboration on this effort. We wanted to open up the conference with a brief intro of both of our organizations, Advancing CTNNB1 Cures and Treatments, which we refer to as ACT, and CTNNB1 Syndrome Awareness Worldwide, which is referred to as Seesaw, and what our missions and goals are for the community. So I'm going to start by introducing myself. My name is Lauren Cochran, and I am the president of ACT. And in our little brief intro into our organization, you're going to hear from each of our founding members, which includes my husband, Sean, and Nicole and Alex Dashiell. We are two sets of parents, just like you, whose children had delays and endured a rather terrifying path of doctors and tests and more tests before receiving the diagnosis of CTNNB1 syndrome. Like you, we were handed a piece of paper from Unique from 2015, told there was nothing we could do except lots of therapy, love, and stimulation, and we were sent on our way. I remember leaving the geneticist's office with my husband and turning to him and saying, "This is not the end of the road. There hasn't even been very much research done on this syndrome. There has got to be more that we can do." So I connected with Nicole on the family Facebook page. We had both recently moved out of New York City and into the surrounding suburbs, and she expressed an interest in research as well. So our journey began with calls to hundreds of research institutions across the country, as well as reaching out to other orphan disease organizations formed by families and talking to anyone that would speak with us. And it ultimately ended in a meeting with Dr. Chung at Columbia University. We sat there in the conference room. It was the first time the four of us had met, and we were extremely impressed with her knowledge and dedication to rare disease. Over dinner that evening, we decided that we were going to make this idea of creating a research organization a reality. And now I'm going to pass it along to Nicole.
In our first year, we have been able to move quickly and have accomplished many things in a short period of time. We obtained pro bono legal work from one of the largest corporate law firms in New York, and were fast-tracked into incorporation and ultimately 501c3 status. We have a diverse board that consists of members across industries and geographic locations who have all been a major asset in our strategic fundraising efforts. With the help of the CTNNB1 community and Seesaw, we have been connected with many doctors and researchers along the way. One of the researchers we initially spoke with was Dr. Michelle Jacob, who we issued our first research grant to. She is an expert in beta-catenin and has spent much of her career studying it. We cannot wait for you to hear about her promising work going on in her lab to help our children. We are thrilled she is a part of our team. Both Dr. Chung and Dr. Jacob sit on our scientific advisory board and have both been extremely instrumental in guiding us through the process to maximize our fundraising dollars. Thank you so much for joining us today. I'd like to introduce Sean Cochran, who will discuss our fundraising success so far.
Thanks, Nicole. So just to give everybody a little bit of insight into kind of our fundraising efforts, as well as what all your donations are being used on, I'm going to give a little bit of an overview here. So since inception, we've been fortunate enough to operate with a $0 cost basis. So what's that mean? It means that every single dollar raised is going to $1 research. So I think it's a really, really great thing for us to be able to do. And so far, you know, as Nicole said, we've had 501c3 status for less than a year now. However, in that time frame, we've been able to raise over $130,000. That was broken down by two fundraising events, one that we held and one that the Shields held, which raised about $100,000, as well as over $30,000 raised from the broader community via your own efforts, whether it be via Facebook or events that you've done, or just donations through the website. So I think it's really important that it's a pretty diverse mix in terms of where these funds are coming from, because we obviously can't do it alone, and everybody's help has been absolutely crucial in this. Going forward, we anticipate that we'll do 2 to 3 kind of keystone fundraising events a year, in addition to kind of the ongoing fundraising efforts from the broader community. And one thing that I just wanted to note is, to the extent that people are doing fundraising events, it's key to ask about kind of corporate matching. That's been a big source of incremental revenue. All right, so now kind of where have the dollars gone to? So on the left, you'll see a graphic just kind of showing the funds raised and the funds dispersed thus far, and kind of the breakdown. As I said, spent over $130,000 raised and nearly $90,000 dispersed at this point. Again, being that we're zero operating costs, all of that has gone to research. The first thing that it went to was our very first grant to Dr. Michelle Jacob at Tufts. She has been able to create mouse models that are representative of the CTNNB1 syndrome in our children. And the second disbursement is currently earmarked for Dr. Jacob. That'll be $50,000, and that'll get more into testing of existing drugs to see how things improve within the mouse models that she built. And obviously, Dr. Jacob's going to go into further detail once we get to her part of the presentation. And finally, that leaves us with a little over $44,000 to put towards future funding of both research and collaboration and testing. So with that, I'm going to hand it over to Alex to discuss our plans for the future and also how we're actively collaborating with Seesaw.
Thanks, John. So as we think about the future, we believe the future brings optimism. Optimism that one day we can find a therapeutic drug to alleviate symptoms, or optimism that maybe one day there's a genetic therapy that could alter the course of the syndrome. And as discussed, we are very fortunate that we have a scientific community that is interested in helping. As we see it now, we have three goals as an organization. The first objective is to make sure that we can continue to provide capital to support their efforts. Our second goal is to increase our ability to gather information and share symptoms and other medical and behavioral experiences. We think this will benefit ultimately our researchers. And then lastly, and maybe most importantly, is to support and learn from each other on our journey. As we think about fundraising, one of the quotes that I found on another website of a similar rare disease or rare syndrome was, "If not us, then who?" And I think that's our mentality. Fundraising has to come from within the community. We are such a small community, and we have to be the ones that start this effort. Our goal is to build a global fundraising organization. We think the success of fundraising will come from the local efforts at the family level. Small dollars make a big difference and add up quickly. As Sean indicated, we have had success to date, and we think by sharing best practices and tips, we can continue to improve and help others fundraise as well. We know that research is expensive and we'll need a budget well in the millions. But again, there's power in numbers. And once we get going, we think that we'll have access to grants, in particular, potentially from NIH, which I think we'll explore further in this call today. And in addition, as we grow and continue to do research, we hope to access dollars from the corporate world, whether that be pharmaceutical or biotech industries. There's also many non-monetary ways to support our collective efforts. We also need volunteers from various different skill sets, whether that be marketing, public relations, graphic design, software programming, accounting, and language skills, just to name a few. So we're really reaching out to the community. We want to be inclusive, and we want this to be collaborative with all the families all over the world. And then lastly, as we continue to unify our community, gathering and sharing information will be critically important. And I think we'll look to set up efforts to do that more efficiently. One of the ways that we think we can better achieve our collective goals will be to have one larger organization. The benefits of that are efficiencies of scale and operational efficiencies. We'll have a consolidated voice to the research community, and we'll have one brand and one community for fundraising. So with that, we have begun to explore merger between the two foundations, ACT and Seesaw. And we hope to accomplish that merger by the end of the year. With that, I'll turn it over to Mariana to speak about Seesaw as well.
Thank you, Alex. It's been a pleasure to work with you all on this conference, and we look forward to our merger for the reasons that you outline. Although we started, and you'll hear a little bit more about this as I speak, our organization as an awareness and bringing families together. We do have a research component named after one of our early board members who unfortunately passed away a couple of years ago, the Abby Loudermilk Fund. So we're looking forward to, as you say, combining our efforts and for a variety of reasons. I want to thank everybody for joining us today. I know we have a lot of CTNNB1 families with us, as well as doctors, therapists, researchers, and of course, Dr. Chung and Dr. Jacob, our special guests. We're really very deeply grateful to you for your work on behalf of our children and our families. Before we move into today's program, I want to give you just a little bit of background about CTNNB1 syndrome awareness worldwide. As I think Sean said, much easier to say Seesaw, so I'll refer to it that way, by introducing you to our board and executive director and what each of them has done for us. I'm Mariana Parks. I'm grandmother of 4-year-old Ford, who was diagnosed at 16 months with CTNNB1, and his little sister Esme, now 18 months, and is neurotypical. We all live near Seattle. I joined the board in 2017 and became president later that year, and still serving in that capacity. The real Seesaw story is told, though, through our founder and first president, now president emeritus, Tony Rose. Tony and his wife Jennifer are parents of Addison, a first grader who was diagnosed with CTNNB1 in 2016, and her little brother Aiden. Tony is a lecturer in information technology and health information technology at Miami University in Ohio. So he naturally turned to the internet to find out more about Addison's diagnosis. After reading the first few studies, and I know most of us have gone through this, that were posted on the web, and 4 years ago, that was a big difference in where research has gone. And joining the family Facebook page, Tony realized that the literature did not really describe Addison or a number of the children whose parents were on Facebook. So what I've come to recognize as the essence of Tony, he decided to create a better resource and proceeded to do just that. He purchased the ctnnb1.org domain and started building the Seesaw website. He immersed himself in how to run a nonprofit organization. He recruited parents to the board in August of that year, incorporated in October, was approved for nonprofit status in November, and earned a GuideStar Platinum Seal of Transparency by December of that year, which is very important for fundraising, especially in the bigger numbers that Alex was discussing. Tony's long-term goal for Seesaw was to provide a path forward for those who have been diagnosed, realizing that not all families have the same access to information about the effectiveness of different therapies. His vision continues to guide Seesaw and our mission to create awareness of CTNNB1 syndrome, to provide information about symptoms, diagnosis, treatments, and therapies, and to connect affected families. And as we merge with ACT to focus more on research into those treatments and therapies, and hopefully a cure in the long term. If you've been involved in any enterprise that operates largely online and on a tight budget, you know how lucky we are to have had Tony's combination of technical background, creativity, and organizational talent, not to mention his incomparable work ethic. So those attributes have given us an outstanding organization that would otherwise have cost many thousands of dollars to create. So Tony, I know you don't like this, but thank you for your continuing and unflagging work on behalf of our organization and ACT. There would not be a Seesaw without you. Our other board members, I had a screen of all of our board members, but it didn't work. So you've already met Lauren Cochran, who in addition to serving as president of ACT, also serves on our board. As our two organizations work toward a formal collaboration, Lauren's participation on both boards has been crucial. We look forward to our continued work with you, Lauren, and to the larger ACT team. Board member Annie Wood and her husband, Chad, live on a farm in rural Illinois with her 4 children, Preston, Jameson, Finley, and McKinley. Her oldest, Preston, was diagnosed with CTNNB1 at age 8 from the Mayo Clinic in October of 2018. Annie's an elementary and junior high school counselor. She joined the board in the summer of 2019 and serves as our vice president. She stepped up on many Seesaw activities, including being our liaison with ACT on this conference, and has done a wonderful job. Annie brings great creativity along with a wonderful optimism and energy to our group. In fact, Annie has come up with a proposal for post-conference networking I'd like to have her to tell you about right now. Annie.
Good morning. I'm Annie, and this is my husband, Chad. And the one thing we felt we were missing out most on not being in New York together was our connection, personal connection to one another. So what I'd like to start doing via Zoom or Skype is holding and hosting some virtual meetups for our parents to get to know each other better. So we will be starting those soon. After we're finished today, we'll be sharing out our information via email. So reach out to me via Facebook and email. And if you would like to be a part of that, we would like to have you join us. Thank you.
Thank you, Annie, and I hope families will connect with you on this very good idea. It will be available after the conference on our website. Other board members, Bethany D'Alberto is, after Tony, our longest serving member on our board. She and her husband, Mark, live in central New York with their daughters, Gracie and Cecilia. Gracie is almost 18, so one of our older diagnosed children, because we were not diagnosable really before the advent of genetic testing. So we don't have a lot of early information on those children. Bethany is our treasurer. She works part-time in a tax office, which is very valuable because even as a small charity, there is a jumble of tax and financial regulations and with potential penalties that you have to wade through. She keeps us on the right path. Patrice Bradley and her husband live in North Carolina, have 3 children. Their high schooler, Alyssa, was diagnosed just 2 years ago. Again, one of our older children with a late diagnosis. Excuse me. Patrice is a newest member of our board, serving as secretary. She is particularly interested in helping to guide families through the maze of doctors and school systems that they face. As a mother of one of those older affected children, Patrice has fought those battles herself, as have most of the families on this conference, and wants to ease the way for others. Bethany and Patrice, by the way, brought Gracie and Alyssa, their daughters, and their families to Seattle last year, where they were participants in the investigation of genetic exome research, or TIGER as it is called, at the University of Washington. The focus of TIGER is to learn more about individuals with genetic events associated with autism, intellectual disability, and/or developmental delay, in order to better describe how different gene disruptions impact behavior in children and adults. We are fortunate that CTNNB1 is one of the 20 or so genes that they study. I believe we have some TIGER staff registered for today's conference, and I'd like to thank them for the good work they do on our behalf. Kayla Caton joined our team in October 2019 as executive director. She graduated from the University of Washington in 2018, then moved to Taiwan for a year to learn Mandarin. She plans on attending medical school. She also works as a research assistant with traumatic brain injury, and the combination of that and her work with us has led to her passion for advocating for those living with disabilities. We're very lucky to have her, and I want to thank her especially for the outstanding work she's done in supporting this conference. The conference, as we all know, was originally meant to be in person at Columbia University in New York, which we were all looking forward to. But as with everything else this year, COVID-19 caused us to move online. We decided to keep the original date because July 25th is CTNNB1 Syndrome Awareness Day, partly because our symbol, the dragonfly, that most of you are familiar with, is also recognized in many countries on July 25th. So in spite of the change in plans, we are very happy to celebrate the research and advances we're making in CTNNB1 syndrome research with you today. We will have a fundraiser this kicking off National Day with the Dragonfly available afterwards. We'll work with ACT on that again through our Abbey Fund and continue making progress in the research field. And now let me turn the program back over to our colleagues, to Lauren, I believe, who's going to introduce our guest researchers. And thank you very much.
Thank you, Mariana. So we are going to be kicking off our conference with Dr. Wendy Chung, MD, PhD from Columbia University. Dr. Chung is a clinical and molecular geneticist and the Kennedy Family Professor of Pediatrics and Medicine at Columbia University Irving Medical Center. Dr. Chung directs NIH-funded research programs in human genetics of autism, neurodevelopmental disorders, and birth defects. A world leader in the diagnosis and treatment of rare disease, Dr. Chung has identified more than 38 new genes for human disease. Dr. Chung, who works in collaboration with Simon Searchlight, which is what the platform we're using for our registry and natural history study, will be taking you through Genetics 101, the clinical presentation of CTNNB1 and registry findings. Dr. Chung truly helped kickstart our nonprofit journey. She gave us the confidence and the hope that we were capable of making a real difference in the lives of our children and all the children struggling with the syndrome. And then you'll also hear from Dr. Michelle Jacob, PhD, Professor of Neuroscience at Tufts University School of Medicine. Her research studies, funded by NIH and the Cure Epilepsy Foundation, are defining molecular mechanisms responsible for changes in brain function and neurodevelopmental disorders, including intellectual disabilities, autism, seizures, and reduced hearing. She is an expert in the beta-catenin pathway and has spent much of her career studying it and its connection to intellectual disability and autism. Dr. Jacob was kind enough to come to and speak at our very first fundraising event. Her passion for this project and expertise in the beta-cat pathway led us to issue her our first research grant. We can't wait for you to hear more about the incredible work she and her team are doing to advance treatments for our children. So with that, I'm going to kick it off to Dr. Chung to start our presentation.
Thanks, Lauren. Let me see if I can get this working. And if someone can just let me know, are you seeing the full screen on the slides now?
Yes, it looks good.
Excellent. Okay, so I'm going to try and cover several different topics that Lauren mentioned. I'll try and demystify some of the genetics and how to understand this, but quickly go into a lot of the questions that people had. So thank you for folks for asking questions ahead of time. I've tried to tailor my talk to the things that would be of interest to you. So we'll talk a little bit about gene therapy, or what you may have been hearing about gene therapy, and put that in terms of a time frame that may be possible. And then also thinking about the clinical aspects, or in other words, what this means in terms of your day-to-day lives with your kiddos or your other family members. So just to put this in context, we have a lot of genetic information. As we've done this, you've gotten to be a member of this club because your child, or your family member, has had genetic testing that's identified the CTNNB1 change. One of the things that's confusing, and understandably so, is that although this condition is genetic in everyone that has it, it is usually not inherited. So in other words, it is not necessarily in the genetic information passed from a mother or the father. It is usually something that starts brand new with the child with this condition. And I'll explain that a little bit more. But one of the important things that comes out of this, and I want to specifically say this for early and as a reminder, For parents, and I'm especially going to call out the mothers, but for parents, there is nothing you did or didn't do that caused this condition, could have prevented this condition. It wasn't anything in terms of an antihistamine you took while you were pregnant or a glass of wine you had before you realized you were pregnant. This is something that happens for all of us when we have children. And I'll just put myself out there. In my children, we've actually done this experiment, and I know that each of my sons has between 100 and 150 new genetic changes. So that's not unique to you. All of our children have new genetic changes. But what's different in your family is that there happened to be one within this particular gene. It happened to land in this particular gene, and that's why we're all brought here together. So I'll just very quickly say that most of you had genetic testing that either looked at a specific set of genes associated with some sort of neurodevelopmental manifestation, or many of you had a test that we call exome sequencing. The exome sequencing looked at a huge amount of genetic information, and with that we took a shortcut to look at the coding regions of genes, or the parts of genes that are made into protein. It looks like a bunch of gobbledygook, but when I highlight or shine a light on the areas of interest, you can see that amongst this gobbledygook is the Gettysburg Address. And that's what we do in the laboratory when we're doing this. To give you a sense of just how minute in some ways, yet how profound this change is, our genetic information is composed of 3 billion, that's billion with a B, alphabet letters. And in some cases, many of you have either seen or know someone with a condition like Down syndrome. That's a relatively large genetic change in the sense that it's like if your genetic information were a set of 46 encyclopedias, it's like having one extra volume of that encyclopedia. So it's a relatively large amount of information. On the other hand, we can drill down and get down to those 3 billion alphabet letters, like reading text in a book. And what we're talking about in your family, as I'll show you in a second, is something very minute, something tiny. It's not even like a volume that's additional or missing. It's not even like a paragraph, but one single letter in many cases, or a very small number of letters. Yet that change, even though it seems small, can obviously have a big impact. So some people ask me, and certainly people address this question, well, when did this happen? And we actually don't know. From the genetic testing that your family member had, we don't know if this was something that was present in the egg or the sperm. We don't know which one of those it came from, or if it was something that happened shortly after conception when the egg or the sperm came together. But importantly, for anyone that may be thinking about having additional children, this is something that very rarely happens again for parents who have had one child. It is very rare that it happens again, but it is possible. And percentage-wise, it's about 1% of families that have had one child will have another child in the future. And in the Q&A time, if people are concerned, we can address how to deal with that, but it's usually not an issue. Furthermore, for siblings or for other distant relatives, aunts, uncles, cousins, this is something that they do not have to worry about. And so it's really only for the parents of a child with CTNNB1 that this is something we even think about. So I'm going to go just very briefly into how you read your genetic test report or how you understand that information that was given to you. And it's important because it'll feed into this next portion as I'm thinking or talking about how we think about gene therapy, or how someday we might think about actually coming up with a cure or a permanent treatment. So when your child or your grandchild or family member had genetic testing, they were reading out the alphabet letters, and I hope you can see my mouse, but they were reading out these alphabet letters of the DNA, these A's, T's, C's, and G's. And in many cases, there was just one letter that was changed. And when you read this out on your genetic test report, as I'll show you in a second, it shows you the name of the gene, and then this gobbledygook here is telling you at the DNA level the address or the position where that change is and which letter was changed. It's not really so critical in terms of understanding right now, but it is something that means a lot to us, as I'll show you in a second, because the type of change that we have actually changes the protein, changes the amount of the protein, and if the protein is made properly. The types of changes, as I'll show you in a second, that most of the individuals in the community have are what we call truncating changes. So in other words, these are amino acids in the protein, the building blocks of the protein that I'm showing here. And for almost all of the variations that we see, it caused the protein to not make the full protein, but to start trying to make the protein and then give up. And when that happens, it leads to a technical process, which you don't need to worry about, but something called nonsense mediated decay, where the cell just doesn't make the protein from that particular chromosome. So for your family members, what happens is even though they have two copies of the gene, it's really as if only one copy of that gene is working, so that they're making approximately half of the amount of the protein. And that has implications for how we might think about treating it. So this is actually not a genetic test report from any of the members of the community, but just to show you, and this is actually a slightly different condition, but oftentimes your report will have this gobbledygook here. And just to take you through what this means when you read a report, it would say something like CTNNB1 here. For how this is inherited for individuals where it could be passed down theoretically. It's what we call autosomal dominant, meaning, as I said, there are two copies of the gene, one that's working, I don't know if you can see my hands, but one that's working just fine, and then one that has a change so that it's not working. And so the technical term that we use for that genetically is heterozygous. In other words, there are two copies of the gene and there's only one copy of the gene with the change. It's usually de novo, that is, it happens, as I said, new in the person with the condition. And usually the classification would be that it's a pathogenic, or sometimes people will say likely pathogenic variant, to classify that this is the answer. This is what's causing the symptoms in that individual. So let me try and show this with a slightly different schematic. What I'm showing you here is, again, two copies of the gene, the one normal copy of the gene here, and then the other copy of the gene here that has this small typo or spelling mistake. And in this copy of the gene, it's making normal protein, but this copy of the gene with the typo, it's actually, we think, as I said, not making the protein. And so there's half the amount of protein that's there. So why does that matter? So some of you have been hearing about things in the news, the popular press about gene therapy. And gene therapy is really an umbrella term that covers many, many different ways of molecularly trying to either correct or add back the gene. And I'll start out by saying we don't have that answer for the community today. It's something that's very much evolving. There are a lot of people working on this, and they're, in a good way, not just working about it for this gene, but it's really being used across thousands of rare genetic conditions. And so there's a lot of, thinking about how this technology can be used across conditions. It's not just for this community, but really thinking about it, like I said, not just for hundreds, but thousands of other rare genetic conditions. So the reason that I made the point that I did about what we call the mechanism, or how this happens, is because that's critically important to think about which type of gene therapy we might use one day. And so I'll give you an example of how at least some of us are thinking about this. So as I said, we have these two copies of the gene, one of which is working correctly, but one of which is probably not making the protein. And so one way that some people are thinking about doing this is to add back the gene that's missing, and specifically adding one copy of that gene that's missing to be able to replace the copy of the gene that's not working. And I can go into technical details during the Q&A if people are curious about this, but there are multiple ways of being able to add back the gene. But the point would be to try and get the normal amount of the protein that's made by adding that gene back. You may have heard about another method that people call gene editing, or CRISPR, or CRISPR-Cas9, but ways of actually being able to go into the genome, go into the DNA in the cell, and being able to literally cut and paste or edit and correct the gene itself. And so I'm showing that here with this red dot. There are ways that we have of doing this. I'll say that we do this almost every day, literally in the laboratory, not in people, but in cells from people. And we go in and, like I said, cut out this particular change where the problem is and paste back in the correct sequence for this. And then ideally that permanently corrects that issue. Some of the challenges in doing this are to be able to get into all the cells where we need to do this, in particular, all the brain cells, and be able to make that edit. This is technology that's still evolving. It's not ready for us to use clinically today. And that's in part because this cutting and pasting or this editing, we haven't gotten to the point that it's 100% perfect. And so sometimes we have what we call off-target effects, where the precision, even though it might be 99% in terms of going exactly to the right spot and making the correction, even if we get these off-target effects where we might make a change, for instance, over here, even if that's only 1%, that's still too much at this point. We need greater precision, we need greater accuracy. So there are new methods that have been evolving. Some of you may have heard about Edit Prime and other improvements that are being made. So I'm definitely optimistic that this will get better and have greater fidelity, but this is something that we still want to have additional improvements before we use it in people. And so this is ongoing work in the field. To give you a sense, though, of how long this takes, this isn't something that happens overnight. I do think we're in a phase of real acceleration in terms of things getting better. But just to say that gene therapy is something that's been going on actually for decades. So it's been started way, way back, even as we started thinking about the Human Genome Project that started way back here. This is something that's been going on for over 30 years. There definitely has been an acceleration, I would say, over the last 5 years. And some of you may know it's something that we're actually using routinely now when it comes to cancer therapy. It's a different type of gene therapy that we think of, but we're even using it for cancer. And I'll show you an example that I think is very exciting for a condition that's actually really been transformed and what I hope will provide some of the foundation for the future of what we're doing. So let me be clear, this is not the condition we're talking about this morning, but it's another condition I've worked on and I think provides some groundwork for thinking about how gene therapy might be helpful. So this is a condition called spinal muscular atrophy. Again, it's a different condition, and I'm not going to go into the details too much about it, but just to say that it's a pretty common condition. We see it in approximately 1 in 10,000 births. And as we started thinking about this, there was a lot of foundation that we had to actually establish before we could go on to therapy. And I'm showing you this because I think these are some lessons we can learn for our community. One of the foundations that was really, really important for us to establish is what the natural history was, what normally happens and happens over time, so that we'd have a comparison that as we started doing clinical trials, we would know what happens without intervention, and we could be able to compare individuals when we started an intervention to know what the difference was. And I'll get into the natural history data that we're evolving, but I want to just emphasize how critically important this is specifically for rare diseases, because what we ideally want to do is not to have to randomize individuals in a clinical trial. For rare diseases, my goal would be to have as many people be able to get access to treatment, but to be able to provide a rigorous scientific study, we need to know what to expect. And we can either do that because we say randomly, you over here, you're going to get the medication. You over here, just by random chance, we pick it out of a hat, you're not going to get medication. And we compare people that get medication versus those who don't, and we see what the difference is, if people improve or don't improve. Now, one other way we can do this is we can say we know historically how kiddos do over time. We know if they get better, we know if they get worse, we know how things change during puberty, but we know what to expect. And if we have good historical control data that's really, really well established, then we can use that as the comparison when we start an intervention. And that way, if it's a safe medication or a safe therapy, we can compare those historical controls to the people who now start medication. And if it's starting to look like medication is beneficial and safe, two very important features, both safe and beneficial, then it allows us to be able to perform rigorous studies scientifically that we can trust and we can use to go to the FDA to get approval for that therapy. So I'm saying this now because this is something that we need to use the time now as people like Dr. Jacob are developing new therapies so that we're ready. We've got everything in place to be able to go forward. One of the things that we did with SMA is we realized that we needed to diagnose more patients. We needed to diagnose more individuals, and we needed to diagnose them as early as possible so that they could start treatment as early as possible. And so one of the things that you all have done is obviously had access to genetic testing. One of the other things we need to do is increase access to genetic testing for more families so that we can get more individuals diagnosed. Again, be able to establish what the frequency of the condition is and the need then, therefore, as companies are developing therapies to say, there's actually a sufficient market. There are enough people out here who would benefit from treatment that it's worth your while investing in making these new treatments. The way that we did this with SMA was to do that initially, and increase awareness, make sure patients got access to diagnostic methods. But then we even went one step further and we offered population-based screening. Every single newborn baby had access to this testing. And I won't say that this is something we have in place now, but there are some of us who are working towards this so that one day we could have something like that available if there were treatment that were available. We now have the ability, and this is in process nationally, that every single baby born in the United States will have access to this testing. It already is available to about half the babies born in the United States, but within the next few years, every baby will have access to testing. And at this point now, we do have FDA approved gene therapy for a one and done, hopefully, durable, sustainable treatment. That is, that a baby can get diagnosed at birth, have an intravenous infusion, even stay in the hospital for just a day or so, and then hopefully permanently treat the condition. Now, I'm not saying this is easy to get to this point. For spinal muscular atrophy, it took us about 15 years to be able to get to that. But the point is that we now know what mistakes we made in some cases. We know how to accelerate this and make things happen faster. And there are many more companies that are developing these therapies in terms of gene therapy to use across rare diseases. So I'm optimistic that it's not going to take us 15 years if we are thoughtful, if we're strategic about how to approach this. I just want to show that this was actually one of my first patients. This is a particular type of spinal muscular atrophy where we diagnosed her within the first 2 weeks of life and she got treatment. This is a condition where she would not, at 14 months of age, either been alive or even able to breathe on her own. But you can see she looks like a normal, healthy toddler, normal strength. And I do think that we have the ability to really move things forward for rare genetic diseases if we're working together. And that's why I'm so pleased to see the community coming together. So let me take a second to pause, and I'm going to shift gears now. So you all know even better than I in terms of the manifestations of what we see with CTNNB1. And I'm going to go over this, but for those of you that may have to take care of your kids or run out for a bathroom break, I'm going to give you the highlights and then I'm going to show you the evidence behind what ultimately is the take-home message. But this is the take-home message, which, to be honest, is not going to surprise you, I hope. So really, this is mostly a neurological condition. Mostly this is something that's above the shoulders is the way I'd like to think about it. So it's something that affects the brain. I think the good thing is, although the data are still emerging, and I still think we could use a lot more data, we're not seeing that it's a degenerative process. Let me underscore, not NOT, not seeing that it's a degenerative process. Our kiddos are continuing to learn, to develop, to gain skills over time. They're continue to make forward progress as we're going forward. And I'm optimistic that as we diagnose kiddos earlier, as we start interventions earlier, and as we come up with supports, technological supports, other better methods of teaching and understanding what's going on that they do understand, I'm optimistic in terms of improvements we can make. I'll show you the data, but this is largely not a medical condition in terms of other parts of the body. So not heart conditions, kidney conditions, liver conditions. It really is mostly a neurological condition, and we're not seeing complications for other things any more so than we see for regular children. And I'm going to specifically say, because I'll get to it at the end, in terms of COVID, or people have been worried or curious or worried about COVID and how it might be affecting kiddos in terms of specific infections or anything like that. Um, I can say that we've done several things to be able to look at this. Number one is that some of you may know New York City was the epicenter really of COVID at the very beginning. And we have extremely, I'd say, extensive experience with children and children with special needs with COVID. And I'm very glad to say that children in general, but children also with neurogenetic conditions like CTNNB1 seem to be resilient to this infection. And so in terms of, I'm not saying that, they can't ever get COVID, but I can say from manning the hospital at the Children's Hospital of New York, we did not, underscore not, see a lot of kiddos coming in sick, even some of them who had this condition that you may have heard of, that's this multisystemic inflammatory Kawasaki-like condition. It's very, very rare, and we can very effectively treat it with standard medication like steroids and intravenous immunoglobulin. So I'll get to why that's important to me at the end, but I want to reassure people that COVID has not had a significant impact on the community here. For those of you who may be grandparents or other family members, I'm much more worried about your health than I am about our kiddos with CTNNB1. So again, I'm going to go through now how I got to the conclusions I'm showing you here, but predominantly this is a neurological condition. So to introduce, Lauren had mentioned earlier, I run a program that's through the Simons Foundation that's called Simons Searchlight. This is something that is absolutely 100% free to the community. And so we've developed infrastructure that helps in terms of understanding rare genetic conditions like CTNNB1. And I want to give everyone the reassurance that this is, number one, completely free to the community. Number two, we've been around for 10 years, and I expect we're going to be around for decades longer. So we're not going anywhere. We'll continue to provide the support. And our mission in doing this is to facilitate research within these rare genetic conditions and to support both on your side, as well as people like Dr. Jacob on the researcher side, so that your investment with the time and the energy you spend participating in research and giving information about your family member can be used by many, many different researchers. So they don't each have to keep coming back and bugging you over and over again. All of your information is private. It's de-identified, so no one knows who you are, but we're able to distribute this information, both the information as well as, in some cases, for those of you who provide blood specimens, we're able to share that with researchers around the world, and all of that is free to them as well. So it helps, I'll use the term de-risk research. In other words, it makes it easier for researchers to do the research that they want to do. If they have good ideas, then it's making it, as my son would say, easy peasy lemon squeezy. We want to make sure that it's as easy for them to participate or to engage in this research as possible, and we want to make sure it's as easy for you to participate as possible. So as I was saying, we began Simons Searchlight about 10 years ago. It was called Simons VIP originally. Some of the folks at UW know. I will say a shout out for the Tiger study, which is incredible. The University of Washington was part of the original Simons VIP study, which we've now called Simons Searchlight. This is, again, not just for one gene, but we have over 100 different genes associated with rare neurogenetic conditions, and we support all of those. So this is infrastructure across conditions. I'll go into the data that we have from Simons Searchlight for this community, but just to let you know, if you do want to join the study, just Google Simons Searchlight. It'll come right up as the top thing on your search. As we do this, and I'll show you, the first step we do is we look at your genetic test report just to make sure that this is in fact the right club that you should belong to. In most cases it is. It's always reassuring though, I think, for families to hear that, but we want to make sure that in rare cases we've had to put people in another category, another club, another gene. After that happens, I have a team of genetic counselors who are both very experienced and very nice, who will set up a time for individuals anywhere in the world. So let me emphasize, this is an international study. The genetic counselors actually speak several different languages, and when we need to, we get translators as well to talk with you, to understand your journey, what it's been like. And it's usually the caregiver of the child we're talking to, oftentimes a mother, but mother, father, anyone who's the caregiver, talking to them about what their child's medical issues have been so that we can understand just what has happened. And then for those of individuals who have been in the study, we do a brief update once a year. So we do what we call an medical history interview and update. In addition to that, we'd like to get some sense of how the children are doing in terms of development. And so this is a parent reported measure that's very well established. It's a standardized measure. So researchers around the world are used to using data exactly like this. And we talk to you to figure out how your child is doing. And again, this is something we do on an annual basis, and it takes a very brief period of time. Altogether, it may take an hour and a half or so of your time to be able to go over this information, and to be able to do updates is usually even less, something like a half an hour or 45 minutes. Those are kind of the basics, what we call the core in terms of what researchers really, really want to know to be able to understand the condition. But for some individual parents who are willing to do that, we enrich it with even more information. And so these are other online measures. So these are things that you can do in your pajamas at 10 o'clock at night after the kids are in bed, but things that you can do that researchers find very helpful to understand the condition better. For those of you who have a child with seizures, to be able to understand the seizures better, as well as some individual behaviors that might be challenging. Okay, so here are the data. So I would say there's good news and there's opportunities. And I'm encouraging and putting a shout out in terms of the opportunities. So here's the good news. Good news is 48 individuals registered with Simon Searchlight. So we know they're out there. We know they had at least found the website and were interested. And here's the opportunity. At this point, only 25% of you have gotten to the point of actually providing that core set of information that I talked about. So that means the opportunity is for the other 75% to get over to this point. So where are the places in terms of where we can move things forward? So about half of the families got to the point of giving us their genetic laboratory report so that we could confirm that they were in the right club. And as I said, 20 out of 21, we confirmed that they are in the right club. So that's good news. Most of you are. But then we had a little bit of drop off here. Individuals who we confirmed didn't quite get to the point of talking to one of the genetic counselors and answering those questions. So those are the opportunities. Again, it's not very much time, about 90 minutes or so talking to people. And I will say one of the benefits that families always tell me is talking to the genetic counselors. If you have questions that are very specific, the genetic counselors either are very experienced themselves and will answer your questions on the spot, or they'll bump up your questions to me and we'll get back to you in terms of things. So it's kind of an opportunity to get some one-on-one time with someone who's really knowledgeable. Okay, so for the researchers, any of the researchers that are out there, people like Dr. Jacob, this is the gobbledygook. And for families, you don't have to worry about this so much, but this is the gobbledygook of the actual genetic changes. In parentheses are the places where we have more than one person with the same exact genetic change. So we have 2 people for each of these 3 genetic variants. And the most important thing about this slide is what I said early on. Almost everyone has a genetic change which interrupts the way the protein is made. So it causes a truncation or probably leads to only half the amount of protein being made. And as we think about gene therapy strategies, this is incredibly important. So knowing and seeing these data are very, very useful, even if families have not gone on to give detailed information about their child. So I thank you for everyone that did that. Interestingly, we do have one missense variant, and although this doesn't happen very often, individuals with different genetic changes can be very important in terms of understanding how the gene is functioning. So everyone matters. One of the other things I will say is that within this, everyone matters. Every single person. And that's because in part it's a rare condition, and that's in part because, as you can see, almost everyone has a different genetic change. we're thinking they act the same way, but it would be helpful to know that for sure. So you have to represent yourself. You can't trust anyone else in the community to represent you or your child. Everyone has to stand up for themselves and be part of this. Okay, so the data I'm going to report to you now are about those 12 individuals who talked with us. One of the limitations is it's only 12. The other limitation is that if you look at the age range of our kiddos, they're age 1 to 12. So that means I can't tell you, based on these data at least, what do people look like when they get through puberty? What are they like in their 20s, 30s, 40s, 50s, 60s? We have a gap in our knowledge that we surely need to fill. So if there are any of you out there, especially who have some of our older folks, it would be really, really important. You can be the big brother, big sister to the younger community so that we can see how changes happen over time. And that is especially important for adults. That's one of our challenges, but one that we're working on. Okay, so I'm going to start with the behavioral issues, the developmental issues. Those are the things that are most consistent. So I'd say basically all of the individuals in the community are having issues, not surprising to you, but in terms of developmental delay, intellectual disabilities. This is not surprising. It is something, as I said, kiddos are continuing to make forward progress. So it's not that they can't learn, but they have trouble learning. It takes more repetition. It takes special education in terms of doing this. Language is a big, big barrier because I think there's a lot that's going on for kiddos, but just being able to be limited in terms of what they can express and tell us makes it challenging. And so I do think their receptive language, their understanding and what they're processing is much better than what they can express back to us. So one thing is don't sell your kiddos short. They understand a lot of what's going on around them, and be careful what you say because they are picking up on those things. There are some challenges and these change over the life course. And so that's part of what we're trying to learn. But some of the kiddos will be diagnosed with autism, attention deficit disorder, anxiety disorder, and they can change both over the life course and circumstance. And so new situations can be more anxiety provoking. And then once you figure out the new normal, things can calm down a bit. As we're seeing this, one of the two things I'm going to emphasize are the senses. So information getting into the brain through the senses, the eyes and the ears in particular. And the reason I'm emphasizing this is because if you can't get the correct information into your brain so that your brain can learn, there are additional challenges. And so we want to be able to take away all of those barriers that are possible. So it's one of the barriers that we can see is the barrier for vision in terms of being able to see the world clearly and be able to understand then what's going on in the world. And so these are all things that are remediable. So we want to be able to find these, see these, and get them out of the way. So the problems that we see sometimes are that there's cross eyes, lazy eyes, but what you're seeing as a parent is that your child is not looking eyes right at you all the time, straight in the eye. If you're seeing a pediatric, and I emphasize pediatric ophthalmologist, if you can, one who's used to dealing with children with special needs is really important because they can't read an eye chart in the same way that, for instance, we can as adults. Sometimes there are problems in terms of vision at a distance or vision up close, and that's where, as I said, a pediatric ophthalmologist can be very helpful. But about half of the kiddos we see have some sort of vision problem that, again, can be fixed, either in terms of glasses, patching, surgery, but something that can be treated. Some of the things we see in terms of manifestations of the way the brain is different is in terms of muscle tone. So it's not exactly muscle strength, although it kind of looks like that, but it's individuals just being loose or things hanging down and not being quite so strong. Very rarely, but sometimes kids will also have high muscle tone, and sometimes that can cause problems with spasticity or tightening, where you need to have physical therapy to keep things loose and limber, or with low muscle tone, you need to have basically a workout, aerobics and working out with the physical therapist to make sure things are strong. Some kids can have problems with movement. So just in terms of gross motor movement, things like walking, running, climbing, and those are things that definitely with therapy can get better. They may be less coordinated, more clumsy, and that poses a safety issue. So we just want to be careful. And obviously this comes into individualized educational programs, individuals who may need to have either special help at school or special supervision. And that's something that I know you guys know about. Some of our kids have been misdiagnosed, I'll underscore misdiagnosed with cerebral palsy or CP. Our kiddos didn't really have problems with not having enough oxygen at birth for the most part, but it really, I think, is before they got the correct genetic diagnosis, people thought it might have been CP. Some of the kids can have tummy issues, gastrointestinal issues. We see this very commonly with neurogenetic conditions. It's frustrating. And sometimes the kids can't tell us when their tummy is hurting, but this can have to do with reflux. This is basically low muscle tone, but in the intestine. And so things come back up. It feels like heartburn, or with constipation. Things just aren't moving through the intestinal system because of, again, that low muscle tone in the intestine. And the easiest way to treat these are with some simple medications, some interventions with diet, and movement actually helps with the constipation. So keeping our kids active. Um, Ear infections, I actually don't think are more common than our regular population. Just, if those of you who have other kids, all kids can have ear infections, but the issue becomes that I find the kids have they just have more difficulty recovering from a cold or an ear infection, and some of them will have repeated ear infections. For those that have repeat ear infections, and my definition for that is 3 infections in 1 year, 1 calendar year, or 1 12-month period, I do find it helpful to put in the ear tubes and clear that fluid out of the ear so that kids can hear correctly. Otherwise, it sounds like you're underwater, and it's hard to hear, it's hard to develop language if you're not hearing all the sounds very crisp and cleanly and clearly. And so this is something that's a simple enough thing to do that if frequent ear infections are an issue, if your kiddos are younger in particular, less than age 5, something to consider. Otherwise, I would say we see kind of the regular things what we see in kids. One person with chronic bronchitis. Again, I don't think it's an immunodeficiency or a primary lung problem, but we do see that. We do see that some things in terms of people having what we call a septal defect or a hole in the heart from one side to the other, or something we call a patent ductus arteriosus. The numbers are small, but I doubt that at this point that this is specifically congenital heart disease associated, but it is one of the things we'll continue to watch. Again, we're not seeing major problems in terms of being short, but we did have one person that had some difficulty gaining weight. We did have 2 of the boys who had issues with undescended testicles. And again, that's something that if they remain undescended, very simple to be able to do a simple surgery to tack those down within the scrotum. So something to watch for. We did have an individual with a tethered spinal cord. So as parents, one thing you can do is look at the back. So your child, if you see something that looks like a dimple along the spine, your pediatrician should have picked this up before, but if you see something like that, bring that to the attention of your pediatrician. That's helpful to know. And then frequent skin condition, especially with allergies, what we call eczema, can be a little red, can be a rash. And I think this is just what we see in general with kids. I don't think there's anything particularly common here. We do see some things that are similar to what we see with other children with special needs. Some kids at some point in their life will have problems with sleep. They may have problems either staying asleep or going down to sleep. Sometimes we see problems in terms of what parents will say is that the extremities, the fingers or toes in particular, may get really flushed and red, or they may get red throughout their body. Or on the other hand, especially if it's cold, they may get kind of blanched or white. And that probably has to do with differences in the autonomic nervous system that we're seeing in terms of blood flow around the body. Some kiddos have a higher pain tolerance. That's something that we also see. And I think this is one of the challenges just in terms of, in general, oral hygiene, is that sometimes dental issues can be an issue. So a pediatric dentist that, again, has experience with special needs children can be very helpful. And sometimes doing things like putting a sealant on the permanent teeth especially can help in terms of giving an extra layer of protection for the teeth if it's hard brushing or hard doing a good job of flossing. Some of the other, or let me say, some individuals use medications. That's a sign to me that something is a significant enough issue to need to use a medication. Allergies and asthma, I think, are pretty just standard fare, but I'm going to point your attention to the ones on the bottom. Again, it's not the majority of individuals, and I do think it may change over time as some of our kiddos are getting to be adolescents or young adults. So I do think this may change, but we do see some need for medications in terms of behaviors in particular. And this last one here, cannabidiol, or is one of the medications that's basically medical marijuana. So where that's been approved now, at least I've seen this for several conditions now where that provides some utility for some individuals. So this is the same slide I showed before, but now you'll just see where the data came from in terms of this. Again, mostly an issue that we see in terms of behaviors. The good thing to me is, again, it's not something where kids are declining, where things are regressing, where children are losing skills over time. I'm optimistic that we can continue to build. But I wanted to end on the COVID note that I alluded to earlier, and I promise I'll wrap things up. I know around the country, different places are having hotspots where things are getting either more or less infected, and we're all struggling with what's going to happen with school with our kids in the fall. One of the things I want to urge you to think carefully about is just the benefit and the risks. And for each family and each household, it's going to be different. But what I've tried doing with my own patients is I've noticed, I've really seen how significant the impact has been on our kids with special needs not to have their therapist. They're not, especially the young children, are not benefiting from Zoom therapy. You really need to get in there. And parents, I know you're doing your best in terms of substituting as therapists, but you and I both know you're juggling a bazillion things, and you're not trained in terms of a lot of this. And I know you're finding it frustrating because we've done those surveys. So I do think if there's some way to safely assess your therapist, either know that they're not infected, and some agencies are doing things like COVID testing or testing for serologies that know people were infected in the past, but probably should not be infected in the future. Figure out a safe way using masks, using hand sanitizers. But I do think the kids have a low chance of infection or problems if they are infected. I think there's a low enough risk to our kiddos and a high enough potential for benefit that thinking of some way to do something beyond just Zoom or remote therapies is going to be important. And for those of you who have young children, especially less than the age of 5, I think it goes double for you all. So within that, think about ways to do it safely. Work with your therapist, work with your pediatricians, your neurologists, your developmental pediatricians. But I do encourage you to think about a solution. And I will say that in the forecasting, I think we've got a window in some places in the country, at least for the next couple of months or so, that getting something in place now is a good time rather than waiting to try and do this at the end of 2020 when things may get a little bit more complicated. So with that, I'll say that there's just a huge team of people that work on this. For any researchers who are watching this, this is the URL to be able to get any of the data, as I said, freely available, and to continue to get updates on this. So go to the Safari-based website, and thank you for your attention. So I'll stop there.
Thank you so much, Dr. Chung, and we'll move on to Dr. Jacob.
Okay, so my screen should now be visible to everybody. And I would like to start out by thanking Lauren and Sean and Alex and Nicole for introducing me to CTNNB1 syndrome. I've learned a tremendous amount from them, and because of interactions with them and more recently with Mariana, I've really gotten very interested in developing a research program around this syndrome. And I'm going to take you on a dive into the science behind a lot of what Wendy Chung just told you about as far as what we see as key features in this disorder. As they said at the beginning, I'm a scientist. I've spent many, many years studying the beta-catenin protein, which is encoded by CTNNB1. And I'm going to give you a science lesson into what we think is going on in this disorder. And our goals are to identify the changes that happen in different parts of the body that give the symptoms that we see, and to develop treatment strategies. So as Wendy said, this is a loss of function mutation that occurs in the gene CTNNB1. It's sporadic, it's de novo. You can't predict it until it's there. Wendy described the major symptoms that we're seeing in your children. And the concept really is that we don't know what are the underlying pathophysiological changes, what is going on in the body to cause these changes. And our research goal is to define those changes, because that's the way we're going to figure out therapeutic strategies. And our plan is to use two different, what we call preclinical models, and I'll introduce you to those ideas and my thinking about drug treatment strategies. So I'm going to go through some of the science. CTNNB1 gene encodes a protein, beta-catenin. This protein is expressed in every cell of the body at all stages of development through to adulthood. It's essential in the body. If both copies are missing, the embryo doesn't even form. You cannot live without this protein. It has core functions. But when I look at the most relevant or most common features in CTNNB1 syndrome, two cell types seem to be major players: nerve cells and skeletal muscle. And so I'm going to tell you some scientific knowledge about what we know right now about the consequences in nerve cells and skeletal muscle when beta-catenin is not functioning normally, when there's a mutation that changes the levels of expression or the function of that protein. And then I'll tell you about the models that we are developing to study particularly CTNNB1 syndrome. And then I'm going to tell you about, there is no treatment for CTNNB1, but there are related disorders that have some similar features. And there are some drug treatments that are being tested in mouse and very small pilot studies in humans that are directing my thinking about how to approach this disorder, and plans that we have now put in place for testing drug treatments. So essentially what our research goals are is to really understand the molecular mechanisms underlying this syndrome. I said we have two preclinical models. This is sort of state of the art in the field. So you want to have an in vivo model, and mouse is a really good model for genetic manipulations. And so we have developed a full body beta-catenin knockout. It's, as Wendy referred to it, as a heterozygote. It's a complete deletion of one copy of the gene in all cells of the body. And we are also working to develop a human-based model. So here it has to be in culture, or we say in vitro. And there's technology now where you can take fibroblasts, like from a skin scrape, or blood cells from a patient, and you can do reprogramming and get them to differentiate as neurons and skeletal muscle. So changes you see in the mouse, you can see if the same changes happen in the human cell with that genetic mutation. And if a drug treatment works in the mouse, will it also work in the human cells? And this is the standard in the field, that you have to have this information to then start to get companies interested, pharmaceutical companies, venture capital, for clinical trials. So if we understand the molecular mechanisms, we can go to points of intervention. And our goal is to get beta-catenin levels back to the normal baseline values. That's the way we're thinking about it, and to have both safe and effective therapeutic interventions. Wendy mentioned you don't want a therapy that could potentially cause other problems. So what do we know about beta-catenin right now? There are several mutations in this gene. Some of them cause loss of function, some cause gain of function. The mutations show a very strong genetic association with intellectual disability. There are mutations that are in beta-catenin itself, the CTNNB1 gene, and also other genes that interact with the beta-catenin pathway. And they strongly link to intellectual disability and autism. So it is a core pathway in these disorders. So I'm going to tell you some of the science now behind what we're seeing. So this is a little stick figure that's a schematic of the beta-catenin protein, the protein encoded by CTNNB1. And I'm just going to point this out because when we have different mutations along the protein, if we want to understand the consequence of that mutation, we have to know where it's occurring in the protein, whether or not a product is made or not. So let me go through a couple of slides to explain the thinking behind this. So this is what we call the amino terminus, sort of like the front end of the molecule. And what's interesting about this part of the molecule, or the protein, is that it has a couple of amino acids that undergo what's called a post-translational modification. It's a change that occurs. It's a phosphorylation event. It changes the charge, which will change the interaction of this protein with some binding partners. And there's a particular post-translational modification that's really important for us to think about, and I'm going to come back to it in the end when I talk about potential drug treatments. There's an enzyme in all of our cells called this glycogen synthase kinase 3. It will phosphorylate beta-catenin in this region, and that causes it to have a higher affinity for proteins that cause the beta-catenin to be broken down. So it occurs normally to regulate levels of beta-catenin in your cells, and there are inhibitors for that enzyme, and the body actually has its own natural inhibitor. And with activity in the nervous system, that causes that enzyme to be inhibited, and it's known that it leads to a temporary increase in beta-catenin levels. And this is a concept, as I said, I'll come back to at the end of the talk when I'm talking about potential pharmacological interventions, drug treatments that could be therapeutic. So in the middle of the molecule are these repeats, these armadillo repeats, and they interact with proteins that are important for adhesion, for one cell to bind to another, which is really important for like keeping barriers, keeping cells intact, like your skin, your intestine. In the nervous system, those adhesions are very important, where one nerve cell contacts its partner and creates a structure called a synapse, which is the structural unit for information transfer. So the cell-to-cell adhesion is very important as well. And then at the end of the molecule here, part of these armadillo repeats, and at the C-terminal, this part of beta-catenin interacts with proteins that cause changes in gene expression. So this is a very, very schematized model of a nerve cell blown up thousands of times, where on the presynaptic side, you have chemicals that are released that activate the adjacent cell, its partner. This is what we call a little synaptic spine. This is the site of rapid information transfer. And beta-catenin function at this synapse is critical. So beta-catenin will interact with adhesion molecules that hold the synapse together, stabilize it, control maturation of that synapse, and actually how many synapses a nerve cell has. So that affects brain function. I mentioned this enzyme, GSK3 beta, that's present in all of our cells together with beta-catenin. When you have activity, not only is a chemical release that can excite or inhibit this cell, but there's also another protein released. It's called Wnt, soluble Wnt. It binds to receptors on the cell, and that can inhibit this enzyme GSK and cause a very transient increase in beta-catenin locally, and some of that will then go into the nucleus and change gene expression. So these are two key functions, the adhesion and the gene expression with Wnt signaling, two critical beta-catenin functions that are really the basic mechanisms that we need to understand how they are altered by gene mutations, because these changes affect the formation and function of the nervous system. So there are approximately 30 different mutations in CTNNB1 that have been reported, and they have all different consequences. I'm focusing on the loss of function mutations. There are a few that also cause gain of function, linked to cancer, and from work in our lab that I'm not going to talk about, also causing intellectual disability. But when we look at these different mutations, and this is the work that people like Wendy are looking at, is what are the consequences of the mutation? They can occur sporadically throughout the DNA. And what happens is many of them will cause what Wendy said, a truncation. You get a smaller than normal product. So we have a sort of an example here, where here's a stick figure of the full-length protein, and then you have a mutation that causes part of that protein to be missing. And what we really need to understand for each of these different mutations is this little fragment made? Does it retain function? Like, can it still bind to some of the critical molecules, like the adhesion molecules, and retain that function? Or in another scenario, it might bind that binding partner, like the adhesion molecule, but it's in the wrong place, and it takes that adhesion molecule away from the one normal copy, and the consequences are worse. That's called a dominant negative. And so we can predict what we think will happen with these different mutations, but we actually have to test it. And that's part of the reason why the different children show different phenotypes. So when I think about CTNNB1 syndrome, I think about what are the critical cell types we really want to focus on. So we look at the core features, and for the learning disabilities, that's going to involve nerve cells in the brain. So we want to look at excitatory and inhibitory neurons and glial cells. For the changes in motor function, that's going to involve motor neurons in the spinal cord and also the skeletal muscle. And for the truncal hypotonia, the postural muscles, these are slow tonic muscles. And then for the potential for spasticity and muscle stiffness, these are muscles that are in the arms and legs. These are fast twitch muscle fibers. So these are the different cell types we're going to focus on. I'm going to show you a couple of tables. I'm not going to spend too much time on them. This is just to show you that there are several mutations in CTNNB1 that have been identified in humans. The phenotypes are all the things that Wendy talked about, that I introduced at the beginning, a lot of intellectual disability and autism. There are other genes that affect the expression of products that interact with beta-catenin, and loss of function also causes these same types of symptoms. That's the human work. Now let's go to mouse models, because the mouse enables us to do experiments where we can get some more detailed information. And I'm going to summarize a bunch of studies that I think are very relevant and are really influencing our thinking. So the first one is work from my lab. So let me point out that the mouse is a model where you can do genetic manipulations, which it's very powerful. You can target your mutation to a particular age or a particular cell type. And so what we did is something a little more drastic than what's happening in CTNNB1 syndrome. We knocked out both copies of CTNNB1 in nerve cells, excitatory nerve cells of the brain, starting from a late embryonic age in the first couple of weeks after the mouse is born. And what we found is that that change caused a learning disability in the mouse, increased anxiety. The adhesion function of beta-catenin was compromised, and there are fewer excitatory synapses. Another group did the same type of mutation, but a later age of development, found very mild changes just in some of the firing properties of nerve cells that would slightly reduce function. So when that mutation occurs is important. Where it occurs is important. One group knocked it out in inhibitory neurons. We were doing excitatory neurons. They found a different readout. They found autism-like behavior, again, increased anxiety. And here, the learning disability was really mild. It was just cognitive inflexibility. And there was more inhibitory function, but they found it actually improved spatial memory. So different cell types seem to be affected differently. A group has studied the skeletal muscle. Knocking out beta-catenin in skeletal muscle actually causes most of the changes to occur in the nerves that innervate that muscle, and it changes the firing property of the nerve that changes the contraction ability of that muscle. So these are knocking out both copies of the gene targeted to particular cell types. I've been, during my time of working remotely, not being able to go into the lab because of COVID, I've been able to do very deep literature searches. And I found that there are some studies where they've actually reduced the level of beta-catenin in cell types of interest. This is a muscle cell line where they reduce beta-catenin levels to approximately 50%. And they find changes in the firing properties of the muscle. And the kinds of changes they're reporting in muscle could cause a muscle to be weaker. And so that's really interesting to us. There's one group that knocked out one copy of the beta-catenin gene only in dopaminergic neurons in substantia nigra. That's a part of the brain that you might recognize because it's involved in Parkinson's disease. And with loss of one copy of the gene, they found that there was more oxidative stress, and that made the cells more vulnerable to future insults that could affect the levels of these neurons, the dopaminergic neurons. And you know, Parkinson's type symptoms can give things like spasticity. There's one study where there's a point mutation, a single change in one amino acid in the beta-catenin protein. And this has been studied in both humans and mouse, the same exact mutation. Just one copy of the gene mutated. The humans show the symptoms you're all very familiar with. What's really interesting is the mouse shows the same type symptoms, and we are able to do molecular studies in the mouse. And what they've shown here is that one mutation will cause a loss of function in the adhesion, but a gain of function in the Wnt gene expression. And there are some structural and functional changes in the nerve cell connections in the brain. So let's summarize the take-home message. One take-home message here is that the same point mutation in mouse and human has very similar pathophysiological changes. And that's important because it says there's real value in getting a deep knowledge of the effects of beta-catenin mutations in mouse models. So I'm going to go through some of the work that I mentioned, but with a little more detail. So this is a paper from my lab that actually this is how we got connected to the CTNNB1 group, because this paper, when it came out, some family members found us, connected us with Lauren, and really got us motivated to start to study this disease. So we were doing either gain of function or loss of function mutations in beta-catenin in the mouse. As I said, this was a knockout where we took out both copies of the beta-catenin gene only in excitatory neurons. And it was a period we targeted this mutation to a stage of the brain development that's critical for synaptic wiring. Lots of synapses are forming at that age. And what we found is that by taking out both copies, this is, as I say, more severe because in CTNNB1, you only have one copy that's missing. What we found is the mouse has learning impairments. And there are diagnostic behavior assays that have been developed for mice that are great for studying things like learning, autism, anxiety. And this is just a little example. I thought it might be fun for you to see one of the types of behaviors we do. It's called the Barnes maze. This table has 40 holes. There's a little tunnel that the mouse can hide in. It doesn't want to be exposed. So we do our manipulations, the genetic manipulations. So for every litter of mice, some of the pups in that litter are normal gene for CTNNB1, and some have the mutation. And that enables us to compare them. So if we did a drug, we could say, does it affect only the mutant, not the normal? It's very important to have a control in that same litter. So those mice in that litter, they learn very well. There are spatial cues on the wall. They use those cues and they learn this is where that tunnel is, and they run right to it. Our beta-catenin mutants have more trouble. They are not able to find that hole, and with more training, they still have trouble. So they have learning disabilities. They have increased anxiety. They don't show autism-like behaviors. And we targeted nerves in the brain. And so motor functions all look normal. We know that the adhesive functions of beta-catenin are dramatically reduced. There are fewer excitatory synapses. If we look at the gene expression function of beta-catenin, we got a surprise. It wasn't reduced. And this tells us there was a compensation. We actually found another catenin family member that gets upregulated when beta-catenin is low. And these type of things you can't predict. You have to do the particular mutation in a model like the mouse and say, what are the changes that occur with that particular mutation in CTNNB1? What functions are lost? What functions might be gained? Because that tells us how we can design a therapy. And there's ongoing work in our lab that is actually finding even new roles for beta-catenin that are really critical for proper learning. So the molecular mechanisms, understanding them, are going to really help us to know the symptoms and how to change them. This is skeletal muscle work I wanted to show you. This is from Lynn Mai's group a couple of years ago. So this little picture over here on the left is striated muscle. You can see the stripes. The green is a nerve coming in, and this big red blob, that's what we call the end plate, the nerve muscle junction. The nerve innervates the muscle, tells it to contract. And what Lynn Mai's group did is they deleted beta-catenin from the muscle, not from the nerve. And what they found is that the signaling from beta-catenin from muscle, those changes actually change the nerve that innervates the muscle and causes changes in the function and maturation of that nerve that would make it a weaker contraction, less excitable. And so, and that comes from the adhesion functions of beta-catenin in this case were not relevant. It was a gene expression change that was most dramatic. So I told you about a couple of studies where rather than completely taking out beta-catenin from nerve or muscle, what happens if the levels are reduced? Because this is closer to the way we're thinking about the effects of CTNNB1 syndrome. So if we have a complete deletion of one copy, we expect about half of the protein to be made. What happens in the body when there's half of the protein? So work has been done in mouse where they've shown that if they remove one copy of beta-catenin or the CTNNB1 gene throughout the entire body, that the mouse goes through embryogenesis normally. It's born and it looks like a normal mouse. They didn't go on to older ages and do all kinds of testing, but they showed that the adhesion functions are sufficient, that the Wnt gene expression is sufficient. So they said, okay, what happens if we have even less than 50%? So they did 25%, 12%. As they go lower, the consequences got more significant. The 12% of beta-catenin throughout the whole body was not sufficient for normal development. And what they found, which is interesting and something I want to pay attention to, is that different tissues require different levels of beta-catenin to differentiate, mature properly, and function properly. And a lot of the changes are in the gene expression aspects of the beta-catenin function. When you reduce beta-catenin levels in neurons and muscle, you see changes in particular functional properties. And these are the functional properties that affect the firing, as we talked about. The muscle weakness in one case, or the muscle stiffness and spasticity in another. So this is giving us important clues as to what is going on. And in the dopaminergic neurons of the midbrain, loss of beta-catenin there makes them more vulnerable to further insults. So what are the key variables? What do we have to really pay attention to? First thing we want to know is for the different mutations, what are the levels of beta-catenin in these tissues of interest? Then for the mutation, what does that precise mutation do? There could be a complete deletion, and we expect to have about 50% levels, and that will get you through embryogenesis without significant issues, and so that's good news. If it's a partial deletion, that raises a lot of questions. Certain changes in sequence make the coding, the mRNA, unstable. Or the protein might not be stable, it might not stick around. So is a protein fragment produced or not? There's a little idea of what it might look like. If there's a fragment, does it retain interactions with key binding partners? That could be positive because it could keep some of the important functions, as we saw with some of the point mutations. Or it could be negative and cause worse consequences because it could sequester that critical binding partner away from the one normal copy. So we can make predictions, but we actually have to test the different mutations in cell lines and see what the consequences are. What are the effects on adhesion? What are the effects on gene expression? That's the bottom line. It looks like it's cell type specific, and there may be age-dependent differences. That's going to be really important. So we want to really focus on progressive changes and compensatory changes over time. Some of them have been seen in mouse studies from our work as well. And one of the questions parents ask is, if we have this mutation, why do the kids look so different? So even with the same gene mutation, same mutation in CTNNB1, there are other factors. There are other genes that have variants. Wendy mentioned her own children that had multiple variants. Those genes, if they interact with beta-catenin, you could have different outcomes with this one mutation in beta-catenin with different variants. Also, environmental interactions, what we call epigenetics. There are changes that occur that alter the level of expression of genes that don't change the coding, just whether or not that gene is expressed at higher or lower levels. So let's focus more now on CTNNB1 syndrome and what we're doing. So based on everything I've told you, we've gotten some really important insights into what we think is going on, what we want to focus on. But most of this is from targeting beta-catenin manipulations to a particular cell type. And we know that the consequences in one cell type are a little different from the other. You change the muscle, it affects the nerve. To understand this disorder, we need to have one copy of beta-catenin manipulated throughout the entire body. And so to do this, after meeting with Lorraine, Nicole, Sean, and Alex, we got pilot funding and we generated a new mouse model. And this mouse model is now, after a couple of generations of crossing, we have a germline mutation that takes out one copy of CTNNB1. So we're very excited. This is going to be a really important mouse model for us. And what we want to do, and we're starting to do it, is to say, what is the molecular framework and can we test treatment strategies? And this is considered an in vivo preclinical model. And we're focusing right now on this complete deletion of one copy. It's going to be important later on to look at partial mutations, but we're starting here. So I'm going to tell you in a couple of slides really what our research overview is, what we're doing, where we're going. So as I said, we've generated this mouse model now, and we want to see, does it recapitulate the key features of CTNNB1 syndrome? So we were very much slowed down, unfortunately, by the coronavirus. The lab was closed down in Boston for 3.5 months. But I can tell you in this past week, my postdoc did a very quick and dirty pilot study. And we're very excited because we think we have a motor phenotype in this mouse. And we're going to confirm that. We're expanding the colony now. And if that holds up, then we have a great model and we're really going to learn a lot. So we're very excited. And this, as I say, was thanks to funding from ACT. We want to define the molecular and functional underpinnings. We're going to look in brain, spinal cord motor neurons, and skeletal muscle. We're going to look at different ages. The beauty of having a mouse is you can go from development to adulthood. They're young adults at 2 months. They live potentially up to 2 years. And so we can really learn a lot. And then we want to do drug treatment tests. And the critical thing is for it to be effective, but also safe, no side effects, adverse side effects. And what our goal is to bring the beta-catenin levels closer to normal levels. I know from research I'm doing with multiple different mutations in the beta-catenin gene and in genes that affect the beta-catenin levels, that there's a healthy range that's physiological. Going too low or too high causes intellectual disability with multiple consequences. And so that's something we're going to carefully monitor and avoid. We are going to assess the effects on learning and motor skills, and we're going to treat at different ages to see if that's a critical variable that affects the outcomes. So I mentioned that we want to also go to human neurons. You might find something, a treatment that's very effective in the mouse in vivo, which is fantastic, but it may not work in humans. There's precedent for that with other disorders. So it's very important now, it's sort of state of the art, where you have to then see if the same molecular changes occur in human-derived neurons, and then if the drug treatment works in the mouse, does it also reverse the molecular or functional changes in these human-derived neurons? And that's something that if we find that it works in both this in vivo and in vitro preclinical model, that's when you get pharmaceutical companies very excited in working with you and developing clinical trials, which are extremely expensive, and we're going to need that kind of help. We're starting with a complete deletion of one copy of beta-catenin, but it's really important to remember that some of these are partial mutations, and we have our colleagues, you heard from Wendy, Raphael Bernier is here listening in. He's representing Tiger. We have Michael Kruger, Mark Corbett. People are looking at the different mutations in beta-catenin and predicting whether or not, what are the more representative mutations, what are the phenotypes that go with them. And we're very interested in playing a role in helping our colleagues to characterize what the changes would be in beta-catenin function with partial mutations, trying it in the human-derived neuron and muscle cultures we're developing, and seeing if the drug treatment that would work for complete deletion, what would its effect be on partial deletion? So we're open to those interactions. This is sort of, again, proof of principle, proof of concept. This is a poster from a company that I got through an email. And I just want to show you that this is actively being done. I have a few colleagues who are doing this, and we're going to work with them, where you can take fibroblasts, a skin scrape, or blood cells from a patient, and then you can reprogram or edit those genes so that those cells now become neurons and skeletal muscle. And this company has developed motor neurons from humans, skeletal muscle, you put them together, and that's the nerve muscle junction. This is the approach we're going to take. And so now I'm going to, for the rest of the time, talk to you about my ideas about potential therapeutic interventions, pharmacological approaches. As I say, there is no treatment yet, but we have some ideas of, we think, important directions to look in. So obviously the beta-catenin molecule for now is our major focus. And what we want is proof of concept for a pharmacological rescue in vivo. So that's where our mouse model comes in as a critical model. And we're really going to focus on this concept of the GSK3 inhibitors for now, because there's some evidence in the literature that I'll share with you that tells me that that's a very important direction for us to examine. And so there are different syndromes that have some similarities to CTNNB1 syndrome, some learning, some motor deficits that might be related, and certain drug treatments seem to be helping in mouse models, and that's what we are focusing on. So lithium is one of these drugs. Lithium might be familiar to you. It's been used for 60 years as a mood stabilizer to treat bipolar disorder. The fact that it's been used for so many decades is great because we know a lot about its effects in humans. It is a non-selective inhibitor of that enzyme GSK3. So let me remind you that GSK3 will phosphorylate beta-catenin. That causes it to be downgraded, destroyed, partially, temporarily. This is a quickly reversible change that happens in our bodies. But if we inhibit GSK3, which again, our cells will do this with activity, we can do it with drugs, we get increases in beta-catenin levels. Can we take this and use this native protein that's already in our cells, manipulate it with inhibitors and get a positive outcome? I want to stress that GSK has other substrates that it works on, not just beta-catenin. So we always have to be really careful and monitor for adverse effects. But I'm going to show you some data that I personally find incredibly exciting and is motivating me. So it's been shown in mouse models of different disorders that you can actually monitor the levels of lithium such that you can bring beta-catenin back to normal. If the levels were low, you can bring it back to normal. Remember, I said we want to avoid levels getting too high. In rodent models with disorders that have learning phenotypes, so this is Fragile X syndrome and Alzheimer's disease. In Alzheimer's disease, there are many changes, but one of them is that beta-catenin levels decrease with aging, and they are low in Alzheimer's. Fragile X, this is a mutation in a Fragile X mental retardation protein. It's the leading genetic cause of intellectual disability, and it's a single gene mutation that can cause autism. The exciting news is that chronic dietary lithium was able to ameliorate both the behavioral and the morphological changes in the Fragile X mouse and improve learning in both Fragile X and Alzheimer's disease mouse models. There's a muscle model that I found exciting as well. So this is a different mutation. This is a protein that interacts with the Wnt pathway, will regulate the levels of beta-catenin in the nucleus. The mutation, this gene, it's like a model of myasthenia gravis. The muscles are weaker, they don't work as well. And what they found is if they use lithium chloride treatment in this mouse model, treating at embryonic and adolescent ages, so age of treatment, they can go up to adolescent, and they regain the strength in the muscle and it was able to function more normally. So there are some pilot studies in humans with lithium, and I think this is something we're really excited about as well. So these are very small preclinical trials where they have actually started treating children with either Fragile X syndrome or cognitive impairment of unknown cause. And low doses of lithium, children that are age 4 to 11, they showed it's a mild change, but it was a significant, statistically significant improvement in cognitive function. About 20% of the kids did have some side effects, but they were mild. And when they went off of the treatment, they were quickly reversible. So that's good news. About 15 individuals with Fragile X went through a clinical trial. The ages ranged from 6 to 23, so young adults also. And 2 months of lithium treatment improved cognitive performance in one test, hyperactivity, abnormal vocalization, anxiety, preservative behavior. So these things are very exciting. Word of caution, it has adverse effects. It's critical to work with a physician to have routine monitoring of the serum concentration levels and no change in thyroid or kidney function, which could happen. Zinc is another GSK inhibitor. It's actually more potent than lithium. It's been shown to elevate beta-catenin levels in cells, so we can work with the endogenous proteins. This also has the potential for adverse effects. It has several, it's a cofactor for many, many enzymes, so it's really important to carefully monitor the levels and monitor the effects. There's another direction that I want to go with the same concept, but I think it might be more effective. And that is because of these positive outcomes that are happening with these less selective inhibitors like lithium and zinc, many groups and companies are developing what we call next generation, very highly selective inhibitors of GSK3. And this is something we're testing. So we want to correct the beta-catenin levels to become back to normal. We're going to test a couple of these inhibitors. We're going to find the drug and the lowest dose that's effective for this correction. We're going to test age of treatment as a variable. And then any remedy, even partial, would be a really important advance. And we're going to test the absence of adverse effects. And all of this is possible because of this mouse model that we have now. So that's an overview of what we're thinking. We're learning a lot about the consequences of beta-catenin dysregulation. What are the effects that it has on molecular or functional changes? And we have ideas for drug treatments for how we are going to attempt to correct the phenotypes, and we are hoping to learn more. I wanted to introduce some members of my lab. So the very bottom picture is Rob Wickham. That was the paper on beta-catenin knockout. He's now an assistant professor in his own lab. And top here, on the left is John Alexander. John Alexander is a very talented postdoc who works with me on all of these beta-catenin studies, and he is actively involved in the CTNNB1 syndrome project. He has a niece with Rett syndrome. When he was in high school, he cared for a boy with cerebral palsy. He's highly motivated and he's a terrific scientist, so I'm very happy to have him. And these are other postdocs working on other functions of beta-catenin and its effects in the brain. And these are funding. We always acknowledge our funding. I've been funded by the National Institute I am funded by the National Institute of Mental Health, Neurological Diseases and Disorders. I mentioned that I was funded by a pilot project from Cure, and we're getting a fabulous another installment, which helps us to keep things going. And we have been funded by Cure Epilepsy, and we have collaborated with a pharmaceutical company to test some of their drugs in some of our mouse models. So we're very excited to move the field forward with CTNNB1. And I'll stop sharing.
Thank you so much, Dr. Jacob and Dr. Chung. And I am now going to pass it along to Kayla, who is the Executive Director of Seesaw, who's going to handle the parent Q&A.
Hello, everyone. Just sharing my screen here. Okay, so we asked upon registration for people to submit any questions prior, and we had several responses. The researchers did a pretty good job of answering and addressing those throughout their presentations. So I believe this was addressed by do you have anything else to add, Dr. Jacob, about this question?
So this is something, I mean, L-DOPA is used to treat Parkinson's disease. It helps with spasticity. I've seen it can also be used in strabismus, but I feel like for medical questions, I would like to defer to Wendy, who's an MD and would have much deeper knowledge than I would.
So I'm sorry, for me, your question was wonky and I couldn't hear it. Could you repeat the question?
What are your thoughts on using L-DOPA for our children? Talking to the neurologist, there sounds like the only risks are related to GI. And are there any supplements or medications that are recommended for our children?
So anyway, so I'm not sure exactly what symptoms we're trying to treat with the L-DOPA. I don't know. It's kind of hard to do this in that kind of conversation with this. It's not generally something I guess I've thought about for this in terms of most of the symptoms I know about for the children who have this. So I'm not sure. There may be some specific thing that the neurologist was thinking about. In terms of supplements, I would say there's not a specific supplement. I don't know of any supplements that are harmful in terms of multivitamins, carnitine, things like that some parents do, immune boosters that some people are worrying about in COVID circumstances. I think all of those that I've seen generally that are over the counter are fine. I don't know that they're doing anything miraculous, but I think they're fine in terms of from a safety point of view. The biggest thing I see is sometimes in terms of gastrointestinal symptoms, kids may be either due to their preference or other things on a more restricted diet. And so it may be helpful to supplement them with a multivitamin. If they're lactose intolerant, not taking so much dairy or milk or things like that, important to get calcium and vitamin D in. We have seen some reports of low bone density. So that's something I am watchful for. But if kids are, eating a pretty varied diet, lots of fruits and vegetables and things like that, I'm less worried about the need to supplement.
All right, thank you. I also want to remind everybody, so for those of you who are participating in the Zoom, there are, at the bottom of your screen is a Q&A button. And if you click and open that, there should be an answered section. And Dr. Chung has done a great job of answering a lot of questions in there. So check that out if you haven't already.
I'll just, if I can say that one question that came up many times in the chat was trying to think about the age of the children and when therapies are available, who's going to be helped based on the age. And I'll just say that we're so early in this whole process that there's a lot that we don't know. The thing that's incredible about what Dr. Jacob has been doing and having a mouse model, I hope that will recapitulate, at least to a certain extent, the symptoms and the challenges we see in the children, is that we will be able to use, and she'll be able to use those models to test out some of these questions. And so we don't know the answer yet. Is this going to work for children who are up until age 13 or up until 30, or we just don't know. It's too early. But we do have testable ways to answer the questions, and we're going to need to do that. And of course, as we think about therapies and moving things to therapies, we're going to be very careful about safety. And so I won't go into the whole sort of explanation of this, but one can use even just kind of regular mice for safety to be able to test safety. And if some of the same things are used for other purposes. So Dr. Jacob had mentioned lithium. We have a long history of experience with lithium. We know the safety profile. And so that's something that I think we generally feel more comfortable because there are less unknowns about it. We know what the safety issues. We may not know the efficacy issues, but it's important that we do no harm, that we don't do any harm to the children. And the same thing will be true when we think about whether it's stem cell therapy or gene therapy. I think we'll be able to get some of the safety profiles, not just from our community here, but as it's used for other communities and other indications, we'll have a sense of what are the safety issues and where do we need to be careful. So we'll be both looking internally, but externally as we're developing treatments.
I'd like to also answer. So I see several questions asking me about the lithium. Again, I'm not medically trained. I got my PhD in a medical school, but I am not qualified to recommend any particular drug or any dose. I feel that, if it's something you're very interested in, you should certainly have a conversation with the physician treating your child. I am going to test it in the animal model. I'm going to test the more specific GSK inhibitors in the animal model, how long you need to be on, all these things we don't really know. And as I was trying to point out, it could be mutation specific because in some cases we might not actually want to increase the levels. Like when there's a reduced level of beta-catenin, absolutely. When there's a mutation, I would like to see if it's a partial deletion and a truncation, what are the effects of lithium in that situation? So it's very hard to think that necessarily one treatment will be perfect for every different mutation in CTNNB1. And I'm always cautious. I like to have information. I would want to test it experimentally in cell lines and get an answer to that. Somebody asked me if I have interactions with pharmaceutical companies. Yes, I will not disclose which particular company, but I did test some drugs they are working on in our mouse models, and they are aware of what we're doing with this project, and they want to know more, and they're keeping in touch with me. So we do, and this is a major pharmaceutical company, so there is some interest, and I will keep pursuing it as much as I can. So I hope that answers the questions about lithium for everyone.
If I can just amplify one thing Dr. Jacob was saying and I alluded to, is that it really is important for everyone to represent themselves in the community, that we can make guesses about the genetic variants and how they're acting and what they're doing, but we don't know until we actually test some things. And so I'm just putting the offer out there again for the community because this is free. So this doesn't cost you anything. For anyone who is part of Simon's Searchlight, they can, and we have this set up to be able to go to a local place where you can get a blood sample drawn. Dr. Jacob will then be able to use the cell lines, directly test what we're making some assumptions on, but all the things she's saying about, well, this mutation may act this way and another mutation will act another way. You can be represented in her studies, but you've got to do your part. You've got to make the contribution and put your information in, and ideally give a blood sample for those things that she's talking about. But at Simon's Searchlight, we're very, very much dedicated to make sure that we'll fund all of those things behind the scenes, make the cell lines available to Dr. Jacob. And so, but we can't do that. We can't make that contribution for you. So, but we will try and make it easy. And if you can help tell us ways that we can make it easier for you, please do share those tips.
Thank you. So I think this kind of leads into this next question here, which is alluding to what can we do as a community to help progress and help children in an expedited way, as well as what can we do to help research and what to do to help find treatments and things to help the kids with CTNNB1. So besides participating in things like Simon's research, is there anything else that parents can do to help keep things moving forward?
Well, I can mention two things that come to mind instantly, and one is the fundraising. As Lauren, or I think Nicole said at the beginning, research is expensive, and the more resources we have, the more we can take on. It will help my lab, it will help other labs that are getting interested. Things will move faster the more people that work on it. I think this conference is fabulous because the more we also talk to each other and learn from each other, like all of the families that shared information with me that I'm still looking into in detail about the mutations in their children, the symptoms they see, this is helping me to understand how the different mutations might be acting in the body. It directs our research, and I want to thank you for sharing that. And I'm hoping, as Wendy said, as we learn more and we learn what the different mutations are, we get more of a sense of really how to treat it and how to, we call this stratifying. Some people will need something slightly different than others, but we'll be doing the smart kind of treatments that will be most effective.
Thank you. And like I said, some of these may have already been touched on, but do we know if CTNNB1 is a progressive disorder?
So I was trying to say this. I obviously didn't say it very well before, but no. The bottom line is no. It doesn't look like it's progressive. It doesn't look like it's regressive in the sense that people are taking steps back or losing skills or declining over time. The limitation, though, as I said, is that most of the information is derived from young people who are still growing and developing. And so I don't really know what the condition looks like when people are in their 50s, 60s, or 70s. All of us decline over age, so I think that's going to be similar for individuals at that age too. But so far, it does not look like a degenerative or declining or regressive condition.
Thank you. And I believe you answered this in the chat about that for specific disorders that are not associated with CTNNB1, people should talk with their physicians for other risk factors.
So one question came up, I think this was around COVID issues. Everyone's an individual person, and I'll say this is true of COVID or anything that people are asking. For most of you, I don't know your children, and so I want to be clear that I can give some sort of general guidance and I can share with you the information that we have, but please do talk to your doctors about individual either medications or whether or not, you should be careful of something during COVID or other things. They know your child best, and hopefully this will give some guiding principles. I will say all of the information that we've presented is publicly available. You guys are going to be posting it, is my understanding. So if your pediatrician or neurologist wants to see any of this, all of this information is available both to researchers, but also to the people taking care of your families, your children.
Thank you. And I believe Dr. Jacob touched on this in her presentation. But when can we use base editing in the CTNNB1 mouse models? And has it been used in animal models already to correct mutations? And when can we test this technology in our animal models and then our children?
So this is a really exciting new version of gene editing that's going on, where it's particularly going to be effective where there's a single mutation in the coding that causes one change in amino acid. So this company, Beam Therapeutics, is developing this strategy where they can target this particular construct to the CTNNB1 gene, for example, and then just cause an enzyme change that would change the coding to correct that single mutation. And if there are a couple of single mutations, they can do a few in one. This is very much in the experimental stage. It is definitely not at the stage where it can work for humans. It's looking like a very safe, effective, very clever approach to advancing this sort of CRISPR gene editing with potential for less off-target effects, which is great. And they're testing that. It's really important to make sure there are no off-target effects, that it doesn't cause other changes, but in cell lines, in cultures, it looks very, very impressive. Right now, it's being tested in things like sickle cell anemia, where you can take the bone marrow out, manipulate the cells and put them back in. You have to get access. And for CTNNB1, we think we are going to have access throughout the body, in muscle, in nervous system. So targeting is going to be an issue. Immune responses when you introduce a foreign construct. There are a lot of issues that have to be solved. This is several years down the road, but gene therapy is amazing. And this is a very exciting approach. And it's something we should all keep an eye on. right now, the mutations I'm doing in the mouse for now are this complete deletion. We could do particular point mutations. We could see if this could work. You know, there are a lot of avenues we can expand into. So this is going to be something really to keep an eye on for all of us.
Thank you. And so I guess we've kind of touched on this, but is this syndrome theoretically reversible, or was there neurological damage done in the first few years of life?
So based on what I'm seeing from the mouse studies, I'm very optimistic that this syndrome is reversible. In the first few, in the embryogenesis with half of the amount of beta-catenin present looks good. It looks fine in the brain's normal. It's a little bit, pretty much normal. It's a little smaller. There aren't that many structural changes. The kinds of changes that we're seeing with these treatments in different genetic manipulations, you can actually, neurons are very plastic. They can change how many processes they have, how many spines they have. Even in adult stages, it's been shown that there's some reversibility. Some of the functions of beta-catenin that are important for things like learning and memory, these are gene changes and it's levels of gene expression. You can fix that at later stages. So I actually am going into this feeling very positive. And I think the answer to this is, yeah, I think it's reversible. That's why we're doing the experiments we're doing.
And if the liver is targeted for gene therapy and produces the protein our children lack, will that affect the beta-catenin levels in the brain?
Unfortunately, no. So beta-catenin's an internal protein. It's not secreted. It doesn't work that way. So if you correct the levels in the liver, it won't get to the other tissue types. We have to do it with something that will get throughout the body and correct it in the different cell types.
And what are your thoughts on using zinc as a long-term daily therapeutic medication for our children? And do you have any dosage recommendations?
Once again, I'm not an MD, and I always decide that I should defer to the MD in the room. So Wendy, I would not prescribe anything to anyone.
Again, I think zinc is one of those things that what you usually buy as a nutraceutical or is fine if you want to try it. I don't know that it's going to have a profound effect, but I wouldn't go nuts on it in terms of, you know, taking 10 of the zinc tablets that are done. But if you want to use what they sell as over-the-counter zinc supplements, I see people using this more in terms of immune system and trying to boost their immune system these days. Whether or not it does that, how effectively it does that, I'm not sure. I wouldn't expect it's going to have a profound effect, but like I said, I don't think it's going to be toxic.
If anyone does put their children on any of these things like lithium or zinc, you have to work with your physician. They have to monitor the levels because it can have side effects.
Thank you. And I believe Dr. Q&A, but about stem cell treatments. Parents report having huge progress in various genetic diseases. They mentioned improvements in symptoms, ataxia, strabismus. How does this help if injecting 300 plus million cells? And how do the effects last? And which clinic does the researcher suggest?
So one thing I want to say about stem cell therapy, we're certainly not there in humans. We're not there for the brain. There's a real interest in this type of approach for muscle. So muscle is one of these cell types that has multiple cells fused to form your muscle fibers. So they have multiple nuclei. And there's a disorder, muscular dystrophy, where this type of approach is being explored because when muscle gets injured, it has these little cells near it called satellite cells that aren't quite differentiated that can then differentiate, become a muscle type cell and fuse. So if that satellite cell was like a stem cell and it had normal beta-catenin and it fused with your muscle, it could introduce more beta-catenin into a muscle that has less beta-catenin. So I think this is something, as I say, it's not in place yet because they have to find ways to grow enough stem cells to introduce them such that they actually integrate into the muscle and don't cause an immune response. And it has potential for positive effects. It would be restricted, I think, mostly to muscle at this stage. But that's something also that is something we could keep an eye on and see what's happening as the techniques are being perfected, because it could provide some benefits.
Thank you. And we talked about this also, but just to reiterate, how long do you guys think until CTNNB1 gene could be possible for repair?
Is this as far as age of treatment, or how quickly do we think we'll have a therapy?
I believe it's alluding to how quickly you believe there until a therapy.
It's always hard to predict. We're talking years, I think, but you know, I certainly will keep the community updated as we make progress. And you know, I'd love to think that within the lifetime of your children, within the next 5-10 years, that potentially we might have some kind of a drug treatment. It would be fabulous. That's my goal. Will it happen? I hope so.
Okay, so on that, I believe that is the end of the questions. Just one more thing for Dr. Chung. So Dr. Chung mentioned that missense mutation during her presentation, and someone just wanted a clarification on what does that mean?
So a missense mutation, and I showed just one of them, is when the change is just one substitution of one amino acid or one building block in the protein for another.
Okay. All right, and then I think all the other questions would be in the Q&A, but we are kind of running out of time. Let me see if I see anything real quick. I think a lot of these are alluding to specific symptoms in kids. Let's see. There's one person asks, is there more work going to be done about seeing if misdiagnosed children with CP are being tested for CTNNB1 gene mutations?
So there are actually some publications now that have come out that talk about genetic testing specifically for the indication of cerebral palsy. And it's not just CTNNB1, but many of those children were misdiagnosed and have a variety of genetic conditions. So I think increasingly, at least at major medical centers, those children are being offered access to genetic testing, and hopefully this continues to expand.
Okay, and there's one question that I see that you marked that you're going to answer live at the very top of the open Q&A about someone's sister who, the very first question in the Q&A you marked you're going to answer live, and then
I actually
we are
didn't.
running
To be honest, I didn't know what I was doing when I hit that button. Sorry about that. But anyway, to get to that level of specificity, it's hard to do this just as a Q&A without knowing the entire clinical context. So I'm going to have you talk to your provider who knows you, and hopefully they can be more helpful than I can, who doesn't know your sister.
Okay.
Can I quickly answer about some of these questions about cancer? Cancer related to CTNNB1 is when there's a mutation that causes abnormally high levels. These mutations cause lower than normal levels, so there's not much of a cancer risk. But yeah, excessive levels, and that's why with the treatments we want to be sure we don't get into excessive levels of beta-catenin, because it's definitely been correlated with colon cancer and some other cancers. When the levels are too high, it increases cell proliferation and cancer type changes.
Okay, so at this point, I think we've gone through most of the questions that were in the Q&A, as well as all the questions that were submitted prior. So at this point, I'm going to hand it back over to Lauren for closing remarks.
Well, thank you all again for joining us. We're so glad that hopefully next year we'll be able to meet in person, but glad that we got together. And very special thank you to Dr. Jacob and to Dr. Chung for taking time out of their weekend to speak to our community. I just wanted to re-emphasize how extremely important it is that you sign up with Simon Searchlight. This is where researchers will be able to pull data and information about our children's specific variants. So it's critical that everyone take the time and that we have a good representation through Simon Searchlight. So I'd really like to encourage you all to do that. And I'd also like to follow that up with the more money that we are able to raise, the more we are going to be able to move this research forward quicker. So we are 4 people, Nicole and Alex and Sean and I, but in order to really make a huge difference, which many of you have already done, but we need your continued support. And we promise that we will keep you in touch with our efforts all along the way, but we're counting on the community to really help move this cause forward. And for additional questions that keep coming in, feel free to email any one of us and we will try to get them answered for you in the future. So thank you all for joining us today.
Thank you for organizing this.
And thank everyone for their help.
Thanks for having us.
Bye, everybody.